Hi, I am Dr. Aida Başgül Martin. Welcome to my channel. In this video, I will talk about the sarcomere structure and function. If you like this video, make sure to select the like and subscribe buttons below if you haven't done so. If you have any questions, as always, feel free to leave a comment. Skeletal muscles are composed of tubular muscle cells that we call muscle fiber. Muscle fibers contain numerous proteins we call myofibrils. Sarcomere is a complex, contracting, repeating unit of striated muscles. For example, in one muscle fiber of biceps brachii, there are approximately 100,000 sarcomere. Sarcomere is the basic contractile unit of the striated muscle. Two of the important contractile proteins are myosin, which forms the thick filament, and actin, which forms the thin filament. Myosin has, as you see, a long, fibrous tail and a globular head, which binds to actin filament. Sarcomere is a repeating unit between one Z line or one Z disc to consecutive Z line. Sarcomere is a repeating unit again between one Z line or Z disc to another, which is the consecutive Z line. Z lines act as an anchoring point of the actin filament, as you see here. Anchoring point for an actin filament, a zigzag formation, Z disc. Surrounding the Z disc or Z line, we see the region we will call I band. I comes from isotropic. I band is the zone of thin filaments that is not superimposed with the thick filaments. Only thin filament forms the I band. When we look to A band comes from anisotropic, named for their properties under a polarizing microscope, and A band contains the entire length, both thick and thin filaments. Within the A band, there is a pale or paler region we call the H zone. It comes from German heller, brighter, named for their lighter appearance under a polarization microscope. H band is the zone of the thick filaments that has no actin, no thin filament, only the thick filaments. During the contraction of a muscle, this area, H zone, and as also the sarcomere, shortens. Within the H zone, you can see here this tiny section we call M line. The disc, the middle of the sarcomere. M line shows the middle of the sarcomere. M line formed of cross-connecting elements of the cytoskeleton. I mentioned actin and myosin as contractile proteins. However, we have more structural proteins. They are not contractile. So contractile filaments or contractile proteins are actin and myosin. Structural proteins, however, troponin, tropomyosin, and titin. Titin is the biggest single highly elasticated protein found in nature. It provides binding sites for numerous proteins and is thought to play an important role as sarcomeric ruler. And I will show you in the next slide. But when we are in this slide, I wanted to mention to you or point out the sarcoplasmic reticulum. That is the area, calcium, which is very important for the contraction of muscle, it's stored and released from sarcoplasmic reticulum, which is the normal cells has endoplasmic reticulum, but when this endoplasmic reticulum found in the skeletal muscle, we call sarcoplasmic reticulum. Here we go. Let's see, the actin molecule has troponin and tropomyosin. The protein tropopyrazine, as you see, covers the myosin binding sites of the actin filament in the muscle cell or muscle fiber. For a muscle cell to contract, tropomyosin must be moved to uncover the binding sites of the actin. Calcium ions bind with troponin C. When calcium ions released from sarcoplasmic reticulum, they come and connect or binds with the troponin C molecule, which are, as you see, dispersed throughout the tropomyosin protein, and they alter the structure of tropomyosin, forcing it to reveal the cross-bridge binding site on the actin. The concentration of calcium within muscle cells is controlled by the sarcoplasmic reticulum, a unique form of endoplasmic reticulum in the sarcoplasm. The myosin head also binds to ATP, which is the source of energy for muscle movement. Myosin can only bind to actin when the binding sites on actin are exposed by calcium ions. The sarcomeres are what give skeletal and cardiac muscles their striated appearance. In here you see a cardiac muscle, and you can easily spot this is a cardiac muscle because of the intercalated disc. It's unique for cardiac muscle, and also branching muscle fibers. So sarcomeres are what gives skeletal and cardiac muscle their striated appearance. When you look at the smooth muscle, they do not have striation and they do not have sarcomere structure. Let's test your knowledge. Question 1. What is the basic contractile unit of muscle fiber? Answer is sarcomere. The thin filaments of muscle fiber are made up of the proteins called actin, and thick filaments of muscle fiber are made up of the proteins called myosin. So myosin and actin are the contractile proteins of the muscle fiber. I hope you liked it. If you do so, please make sure to click like, share, subscribe button below, and if you have any questions, leave a comment below. Have a good day.